Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. From this class, we shall continue our discussion with the tools of recombinant DNA technology, that is the vectors. Vectors are otherwise called vehicles of gene transfer, because if we want to move from one place to another place, it is of a large distance, then we need a vehicle for fast transfer, right? Likewise, naturally, the cells are capable of uptaking the DNA, but it is a very slow process. So for that, scientists have discovered the special DNA molecules which are capable of transferring the target DNA into the host of our interest. So those DNA molecules which are capable of transferring the genes into a target host are referred as the vectors. Since they are transferring the DNA, they are called vehicles of gene transfer. So there are very broad vectors available nowadays, but we shall discuss the things like from where the concept of vectors have been started. Here, naturally, there are DNA molecules called plasmids in bacteria. Plasmids are double-stranded circular DNA molecules capable of undergoing self-replication. And there are bacteriophages. Bacteriophages are the virus which can infect the bacteria. So these two are capable of transferring or entering into the bacterial cell, and they multiply or they replicate in the bacterial cell. Suppose here the plasmid is present in the bacteria, and on its own it is capable of replicating. So during the replication, it can replicate the genes present in it. Likewise, the bacteriophages. Bacteriophages, when they enter into bacteria, they transfer their genetic material into the bacteria, and there they undergo replication by using the bacterial protein machinery. So that means here also the replication takes place. Whatever the genes present in the bacteriophage DNA will also get replicated. From there, biotechnologists thought, if we are capable of attaching the gene of our interest to plasmid, if we attach the gene of interest to the plasmid, if we attach gene of plasmid to the DNA of bacteriophage, then what will happen? It can enter successfully into the bacterial cell and they multiply. So from there the vector concept has been started. So here, by using these, that is plasmids or bacteriophages, it is possible to transfer the gene of interest to the target host. And to use these as a vectors, either plasmids or bacteriophages as a vector, their copy number is very important. That is the number of plasmids per cell, number of plasmids per cell, or number of bacteriophages per cell. The number of plasmids may vary few to many, that is 10 to 150 number, it may vary. And bacteriophages number is still more larger compared to the plasmid. So how much number of plasmids are there, that much number of target DNA can So that means copy number is also one of the criteria in order to use a DNA as a vector. Which DNA? Whether we are using bacteriophage DNA as a vector or we are using plasmids as a vector. But nowadays there are many artificially constructed vectors available. Artificially constructed vectors are available for gene transfer, because without altering these genes it is not possible to attach the DNA, or it is not possible to select which host is transferred with the target gene. That we call it as a recombinant. Selection of recombinant is also one of the criteria. That is one of the things we should look at after transferring the gene. So by considering all of these, there are some criteria in order to use the DNA or the DNA molecules as a vector. So let us discuss them in detail. The criteria for considering or using the vectors. Which vector is suitable? It should fulfill some criteria. Criteria for using vector in constructing a recombinant cell. The first criteria could be copy number. Copy number. That is, the copy number is nothing but number of traits, or we can call genes. Number of genes the cell carry. That means how much number of the DNA can be obtained, or how much number of the target gene can be obtained from a cell. So that is one of the criteria. That means, consider in a cell, if the cell is capable of producing more number of target gene, so it is our target gene, if it is transferred into the cell along with the vector, it should be produced in a large number, and that large number will depends always on the vector. If the copy number of vector is more, then the copy number of the trait or the gene will also be more. So how much number of vectors are produced, that much number of gene of interest will also be produced. So always we should look at, there should be production of more copy number, so that vector can be used. That vector which is capable of producing high copy number is recommended for gene transfer rather than any other things. And the second thing is the presence of ori site or origin of replication. Origin of replication. So in order to use any DNA as a vector, it should have the ori site. That means to produce copy number, it should undergo replication, right? For undergoing replication, there should be a strong ori site. Ori site you have studied from molecular basis of inheritance. It is the region where the process of replication will start, where the DNA polymerase will binds, and from there the replication will start and produces more amount of DNA or the multiple copies of DNA. The same thing here. In order to use any DNA as a vector, it should have the strong origin of replication, or in short we can call it as ori site. Ori site which can support the high copy number. Ori site is present, but it should support high copy number. It should support high copy number. So that vector is highly recommended to use in a recombinant DNA technology. And the third criteria is the presence of selectable marker genes. Selectable marker genes. So here the selectable marker genes in a vector will helps. They helps in identifying the recombinants after transformation. Transformation is a process of gene transfer. It is one of the process of gene transfer. Okay, here if it is a target DNA, if it goes directly into the bacterial cell in any way, then the process is called transformation. It is the process of gene transfer. Okay, so after transferring the gene into target host, whether the gene is successfully transferred into the cell or not, here the cell is referred as host. Whether it is transferred or not, in order to identify that, we make use of selectable marker genes, and those selectable marker genes should be present in a vector we are using. It is not possible to insert in the cell, so it should be present in a vector. So usually the selectable marker genes used in recombinant DNA technology are antibiotic resistance genes. Antibiotic resistance genes are commonly used as a selectable marker genes. Apart from this, enzyme coding genes are also used as a selectable marker genes. We shall discuss them, how it is possible to select little further. So here the commonly used selectable marker genes are antibiotic resistance genes and enzyme coding genes. So we shall take an example. Here the commonly used antibiotic resistance genes are ampicillin, tetracycline, chloramphenicol, kanamycin. These are the antibiotics name. So those genes which have a resistance towards these antibiotics are used as a selectable marker genes commonly in Escherichia coli. Why we are taking example of Escherichia coli? It is a commonly used bacteria in biotechnological works. That's why we are taking this as an example. So you may be asked with a question in competitives like, identify the antibiotic resistance genes which are commonly used in Escherichia coli. So 4 antibiotic resistant genes are commonly used for Escherichia coli, for which it is having resistance. Okay, Escherichia coli is having resistance. That means in presence of these antibiotics, Escherichia coli cells are capable of growing successfully. Okay, and this is about the selectable marker genes. How we can select, we shall discuss it a little further. And one more thing, in plasmid, commonly used plasmid, PBR322. It is a commonly used plasmid, first artificially constructed plasmid. Here it has two antibiotic resistance genes. So this is a plasmid. It has two antibiotic resistance genes. One is ampicillin resistance gene. R refers to resistance gene, and tetracycline resistance gene. These two are the two antibiotic resistance genes present in PBR322, which can be used as a selectable marker genes. And the selectable marker genes, as I told earlier, they will be helpful in identifying the recombinants and non-recombinants. That is, whether the cell is having the genes of our interest or not. So, once we are conducting a procedure means it is not sure whether the DNA is transferred or not, because everything is not visible. So, for that, the selection is very important. For selecting, we are using either antibiotic resistant genes or enzyme coding genes. So this is the very important criteria in order to use a vectors. So it should have at least one at least one selectable marker gene. The next criteria for using any DNA as a vector is it should have the cloning site. The cloning sites are commonly called restriction site. Cloning sites are commonly called restriction site, at which the gene of interest is ligated. Gene of interest is ligated. So here on a vector, there should be presence of a restriction site. Restriction site is a place at which the restriction enzyme, the commonly used restriction enzyme, can come and cut it. So once the DNA is cut into fragment, then at that place we can attach or we can ligate the gene of our interest. So for that, there is a requirement of restriction site. At least there should be presence of at least one restriction site. For commonly used restriction enzyme. So whatever the enzyme we are using for a restriction treatment, so for that enzyme there should be presence of one restriction site, not more than that. For example, I told in a plasmid PBR322 there is presence of 8 restriction sites for 8 different restriction enzymes. So I will take a few. Here there is presence of restriction enzyme or restriction site BAMH1 for enzyme BAMH1, SAL1, and these two are present on tetracycline resistant gene. So this is tetracycline resistant gene, and here it is present PVU1 and PSP1, and it is present on ampicillin resistant gene. So if you observe here, for BAMH1 only one restriction site, for SAL1 one restriction site. So there should not be presence of multiple restriction sites for single restriction enzyme. It will create multiple cuts, so the vector DNA will be chopped. The process will be difficult to continue further. For that reason, there should be presence of only one restriction site for commonly used restriction endonuclease, and that site is commonly called a cloning site. at which it is possible to attach the gene of our interest. That all of these are the requirements for or criteria to consider any DNA molecule as a vector.